Hello everyone, I'm very glad to introduce our recent new research published in iMeta, which is titled A New Evaluation System for Drug Microbiota Interactions. Based on the germ-free animal model of drug metabolizing enzyme gene editing and humanization, we propose a drug microbiota interaction evaluation system and research strategy. It explores how a specific microorganism interacts with a drug in the body, using clinical drug screening, screening of differential pharmacodynamics components, causality of gut microbiota on drugs, and advanced molecular techniques. Individual differences in new drug efficacy are affected by gene regulation and environmental factors. Drug development focuses on gene-protein levels, affecting cellular function. However, less than 5% of drugs make it to clinical use after 10 to 25 years and $1 to $2 billion in trials. Little attention is given to environmental impacts on efficacy. Gut microbiota, a key body-environment interface, reflects adaptive changes. Could using gut microbiota as an evaluation criterion reduce drug development waste? Many studies have shown that gut microbiota is closely related to drug absorption, distribution, metabolism, and excretion. Gut microbiota metabolites can function as drugs regulating physiology, while also influencing drug pharmacodynamics through enzymatic reactions, thereby impacting clinical efficacy. Additionally, drugs can alter gut microbiota composition and colonization patterns, and interact with microbiota genes, including antibiotic resistance genes. The environmental factors that commonly affect gut microbiota mainly include four aspects. The first aspect is the impact of the natural environment on specific bacteria in the gut. The second aspect is the impact of environmental pollution on intestine and human health. The third factor is mainly psychological factors and stress, among which psychiatric factors are one of the most important influencing factors in drug metabolism. The fourth aspect is the intervention of individual lifestyle on gut microbiota. Due to the complex interaction between gut microbiota and drugs in the body, current research faces limitations. Therefore, to solve the problems in drug microbiota interactions, we have explored some new methods. First, we use a preclinical drug research strategy based on germ-free animal models, colonizing germ-free animals with single, functionally distinct microorganisms to provide direct evidence for the involvement of gut microbiota in drug metabolism in the body. Secondly, we also screen and design drug efficacy and toxicological components related to gut microorganisms based on the serum pharmacology of precise germ-free animals. Subsequently, we study the causal relationship between flora and drug metabolism based on microbial sorting, cultivomics, and precise colonization of germ-free animals with single bacteria. In addition, we combine the strategy of single bacterium identification and classification with cultivomics to provide important technical support for the role of gut microbiota in the efficacy and safety of clinical drugs in the body. We can also use the 3D in vitro cell culture system of organoids as a powerful tool to study drug effects. This type of organoid is also an excellent medium for co-culture of host cells and microbes. In summary, in this study, we explored the combined effects of genetic and environmental factors on gut microbiota, as well as the significant impact of gut microbiota on the microenvironment of drug absorption, distribution, metabolism, and excretion of the host. This highlights the necessity of establishing a new evaluation system for drug microbiota interactions. And in order to study the complex nature of the interaction between gut microbiota and drugs in the body, this paper proposes a new top-down research model using precise germ-free animals.